Hello, ladies and gentlemen. Let's dive right into Chapter 11, Section 1, Basic Patterns of Human Inheritance. Our essential questions: how can genetic patterns be analyzed to determine dominant or recessive inheritance patterns? What are examples of dominant and recessive disorders? And how can human pedigrees be constructed from genetic information? We only have 3 vocabulary terms for this section. One of them is a review, so go ahead and write those down in your notebook if you haven't already. Recessive genetic disorders. A recessive trait is expressed when the individual is homozygous recessive for that trait, meaning they have 2 recessive alleles. Those with at least 1 dominant allele will not express that recessive disorder. An individual who is heterozygous for a recessive disorder is called a carrier. So they have 1 allele that is defective, but they don't express it because they have the dominant allele that is normal. Cystic fibrosis is a recessive genetic disorder. It affects the mucus-producing glands, digestive enzymes, and the sweat glands. This causes chloride ions to not be properly transported out of the cells of a person with cystic fibrosis, and basically they have too much mucus that they can't break down. It causes mucus excretion that clogs the ducts of the pancreas. It messes with digestion, and it blocks respiratory pathways in the lungs. Basically, the airways get clogged with mucus. Now, people with cystic fibrosis, they have 2 recessive alleles that cause this disease. Okay? It is possible for a person to be a carrier of cystic fibrosis and not even know it. All right, albinism. You guys have all heard of albino organisms. It is caused by altered genes that result in the absence of the skin pigment called melanin. That's what gives your skin and hair in your eyes color. People with albinism, they don't produce those pigments, so they have very, very pale skin, white hair, and pink irises because they are unable to produce the pigments that cause those colors. Tay-Sachs is a disease that is caused by the absence of an enzyme responsible for breaking down fatty acids, and so those fatty acids accumulate in the brain, causing it to not function properly. Children with Tay-Sachs usually progress normally for the first few months of life, and as these fatty acids begin to build up in the brain, they start to regress or go backwards developmentally. So they may learn to walk, but then soon lose the ability to do that. Galactosemia is a recessive genetic disorder characterized by the inability of the body to digest galactose, which is a milk protein, so they can't drink milk products. They can't have dairy, so it's similar to lactose intolerance. Dominant genetic disorders. They're a little bit different because if you have 1 dominant gene, you're going to have the disorder. Huntington's is an example of this. It affects the nervous system, causing gradual loss of brain function, and it occurs in about 1 out of every 10,000 people in the United States. Achondroplasia causes small body size and limbs that are comparatively short, and it is caused by an abnormal gene that affects bone growth. So these are disorders that if you have 1 allele, you're going to have the disease. There is no masking it. If it's there, you're going to have it. And pedigrees. Pedigrees are diagrams that trace inheritance of a particular trait through several generations. Most of them have a key to help you understand what means what. Females are usually circles and males are usually squares. People that are carriers are sometimes marked half and half. A straight line across, like right here, indicates a marriage. A line down from 2 people indicates that they had children. Okay, and we number the generations with Roman numerals. So this is the 1st generation, and this is the 2nd generation. And we number the individuals within those generations with regular Arabic numbers. Analyzing pedigrees. Pedigrees can be used to examine both recessive and dominant genetic disorders. They can be used to trace eye color or the presence of a hitchhiker's thumb. It doesn't have to be a disorder. Information about individuals' genotype can be inferred from the phenotype of the parents of that organism, of that individual, and by the offspring of that organism. It's not 100% because sometimes an organism might be heterozygous dominant or homozygous dominant. There we go. Sometimes it's not known because we don't have enough offspring to look at or enough parents to look at. So sometimes it's a little iffy, but we do our best. Inferring genotypes, knowing physical traits can determine what genes an individual is most likely to have. And predicting disorders. Record keeping helps scientists use pedigree analysis to study inheritance patterns, determine phenotypes, and ascertain genotypes. So if two people that have cystic fibrosis in their family, if they get married, they both have it in their family, they could be carriers. So a lot of times they will go to a genetic counselor who is a scientist to help them figure out their risk of having a child that has cystic fibrosis. So we're going to talk more about this in class tomorrow. If you have any questions, let me know.